core. Now the calcium pumps in the SR, lots of them, which take calcium back up to reduce the calcium concentration. Now just to repeat, I don't remember if I pointed this out last time or not. Calcium can actually work in this state to prevent actin and myosin binding. So elevated calcium, because of the way it works on troponin and tropomyosin, can actually block this actin binding instead. So basically, you must go back and say, well, in actual fact, all these myosin heads have ADP and an organic phosphate bound to it, and it's just waiting to be activated. So if you elevate calcium, you get actin binding, and the phosphate cycle will work. So calcium is an important messenger in many processes, and it certainly is in muscle contraction. Hormone, release of hormones, transmitter release, and it's also a trigger mechanism for muscle contraction, all kinds of muscle contraction. So we went through this the other day. So we know where calcium is stored, how it's released. We'll go into a little more detail on that. And we know the importance of the sarcoplasmic reticulum. Just to point out, in fact, you have a muscle cell, you have myofibrils, for example, that are close to the surface, and some that are sort of deep inside the muscle cell. So the question is, how is this calcium released synchronized? It has to be synchronized very quickly in order to get the fast contraction that you get by that is accomplished by skeletal muscle. The answer lies again in the structure of skeletal muscle. As we went through this the other day. So another structure must actually synchronize the release of calcium from the thermal cisternate of the SR. And this is accomplished by a duct system called the transverse tubular system. The T tubules are extensions of the surface membrane, so they're the same as the plasma membrane. So they're extensions of the surface membrane. And you enter that junction of the AI bands and man. In frog, it's the Z line. So it depends on the species to some extent. And they carry ions, nutrients, and action potential. This is the biggie deep in the muscle cell. And they're coupled with something called aromatase receptors, which are calcium release units. It's located on the sarcoplasmic reticulum. I'm going to show you some pictures when we get into cardiac muscle, because they're bad pictures what the cardiac myocytes. Okay, so that's the SR, or the T system. And if you look at the plasma membrane, the skeletal muscle cells, you see these little holes. And that forms the transverse tubular system, and it actually goes deep into the muscle cells and surround all the individual myofibrils. So that then the action potential travel over the surface membrane. And they actually penetrate the cell and go deep inside the cell and close to the SR or thermal cistern of the sarcoplasmic reticulum. So it's all done by action potential. And they go deep in the cell. And this is the structure that actually synchronizes quick release of calcium from the SR in skeletal muscle. And then basically says it's an action potential travel from the inside before that's the surface membrane, and also travel down the T system or this duct system like you have in your home heating system, all the furnace ducts that go throughout the house, and it couples the surface membrane to the sarcoplasmic reticulum. So it does it very quickly. That's the trick in skeletal muscle. It has to be very fast. So that's the transverse tubular system. Now, this is sort of the large, a lot of information on the picture that I showed you earlier, showing you the terminal cisternae of the sarcoplasmic reticulum on both sides, and the T system, the transverse tubular system. So it's very close to these little storage units of calcium in skeletal muscle. So this is SR, and this is also SR. So the action potential travels down this guy and he polarizes this membrane. Located on this membrane are a type of calcium channel called the L-type calcium channel. Does Dr. Noy mention L-type calcium channels in any aspect? Well, quite similar to sodium channels. The only thing is they're really slow, much slower than sodium channels. And when we get into cardiac muscle and cardiac mechanisms, we'll be hearing about calcium channels a lot. These guys, of course, there's calcium inside here because it's the same as the outside, about 1.8 millimolar calcium in mammalian cells. And inside here, calcium is very low. So inside the T system, calcium is very high. When these guys are activated, they can pass calcium into the cytosol. The only interesting aspect is it's not sure that you actually need calcium in skeletal muscle. No calcium has to go in. So you can zero the calcium in skeletal muscle, and skeletal muscle will still contract. That's contrary to some of the other types of muscle. So, there is some evidence that there is when these things are depolarized, they're gated. You've heard of gating, I'm sure. And the gating somehow or other activates the calcium release units on the SR. They call ryanodine receptors for the fact that an alkaloid of ryanodine actually opens them up and eventually all the calcium is drained. So they call it that because it binds to these guys and opens them up. And that's a pharmacological tool. So you have the action potential comes down the T system, depolarizes this membrane and activates the calcium channel. You don't need calcium to activate skeletal muscle. But they do gate these things, these calcium release units, and the actual coupling between these guys is controversial. This slide says it's a mechanical coupling. It could be. But there's also a chance that it's sort of a charge transfer where these things are gated. So it opens these guys and calcium floods into the cytosol, the myofibrils, and causes either the activation, and when it's reduced, it drops the calcium concentration, pump back up into these guys here to reduce calcium levels and get muscle relaxation. So let's get all the information on there. It's very complex, but the idea is that you have the T system have a specific type of calcium channel, and the sarcoplasmic reticulum has calcium release units which are gated by these calcium channels. Whether it's mechanical or a charge transfer is controversial. I don't know for sure. I don't think anybody does, but this textbook, like the mechanical concept, may be true. But also, a couple of my colleagues have measured charge transfer and things like that. So the coupling mechanism here in skeletal muscle is not clear. But anyway, these guys have to be activated to activate these release units, calcium release units. So that's the way it's synchronized. So, what are the sort of steps that might occur? You have an action potential in the surface membrane, which is generated, of course, by a nerve, like the end plate, get an end plate potential, polarizing the cell to the threshold, you get sodium turning on, you get action potential for propagating. They propagate in both directions from the end plate, And in addition to that, you go down the transvertebral system. You activate these L-type calcium channels on the tubular membrane, which induces calcium release from the SR via these aromatase type receptors with calcium, or you can call them calcium release units. So when they open up, a ton of calcium comes out of the SR. And that's their function. Calcium binds to the component, the TNC, the calcium binding site of the component. You get a conformational change in the component, and this shoves the tropomyosin out of the way. You get actin binding, inorganic phosphate, or PI is released, and you get contraction. Now, once this happens, once you get the calcium increased, the pumps in the sarcoplasm in particular start to work. They're ATP dependent, and they start to pump calcium out of the myocardial to be stored back in the SR. Calcium is less than 10 to the minus 7 because of this pump. It's an ATP dependent pump. The tropomyosin component back to the original shape, tropomyosin slips back, and you get actin binding and block, you get relaxation. So that's just roughly the steps that you would get. So muscle contracts and then relaxes. And all this stuff requires ATP. Adenosine triphosphate is the immediate source of contraction, the energy for contraction we pointed out earlier. And the source of ATP is lipid and carbohydrate metabolism. Now, in most types of skeletal muscle, in addition to this ATP, there's a supplementary source of energy. It's called creatine phosphate. And you have about 8 times CRP in muscle, in most types of muscle, as you have ATP. So you have a storage account of ATP. So when you use up ATP, it converts it to ADP, and the creatinine is phosphorylated, and these two together will generate some ATP. You have 8 times more creatine phosphate in skeletal muscle than you have ATP. The actual or basic effect depends on the type of muscle. Some generate it fairly quickly, some very slow. Now, under severe exercise conditions, using fast muscles which are considered to be fast, or skeletal muscles, for example, running up a flight of stairs. We have a table at the end. You can use glucose directly. Not all muscle can do that, but some types of muscle can. So you can get glucose will generate 2 ATP, one molecule of glucose. Glucose will generate 2 ATPs, and will give you some lactic acid. Lactic acid doesn't hang around very long, it has to be oxidized, it needs oxygen. And so if you see somebody that's done a 100-meter sprint, just one, and they go over to the sideline and they pant, trying to replace the oxygen. That's called repaying the oxygen debt, because of this lactic acid, it has to be oxidized, and that's why you get this sort of great intake of oxygen. So severe exercise conditions in certain types of muscle, for example, running up a flight of stairs, running down a football field and all that equipment, a nice sunny day. These guys all come to the sidelines and they're breathing very hard to try and oxidize this excess lactic acid. So there are several sources of energy for muscle contraction. Skeletal muscle is bad for that. You need to, but the worst type of muscle we'll talk about is, of course, cardiac muscle, because it tends to be a real hog for ATP. The heart's beating 60 times a minute roughly, so it's going away. So you have to generate the ATP. skeletal muscle is, but cardiac muscle is worse. Yeah, 30, 60 seconds or so, we get contraction. Okay, contraction in muscle, the skeletal muscle, is the active process of generating a force or a displacement. And everybody that's taken physics 30 knows that muscle only works by shortening. You can't push a rope, physics 30 sort of thing. So in order to generate any kind of effect, muscle has to shorten. So it works by shortening. Tricky course for physics 30. Can't push a rope. There are several types of basic contraction. We're going to talk about two of them. This is not we're doing exercise physiology and important other ways of presenting this. The first one is isometric. And an isometric contraction is the length of the muscle is held constant, and the force produced is measured. It should be no shortening of the sarcomeres. It should be just force generated. That's not strictly true because tendons are elastic and there will be some shortening of structure. The other type of basic contraction is isotonic. And basically the load on the weight or whatever you're trying to lift is held constant and the length change is measured or the velocity is shortened. That's an isotonic. So you actually get shortening of structure. Body movements, for example, isotonic, pushing on your car when it's stuck in a ditch, primarily isometric. And that may be something you'll be doing for the next few days if you drive. Okay, so those are two basic types of muscle contractions. So to point out, internal physiochemical events are the same. You get calcium release, you get actin binding, you get contraction. The only difference is you get shortening or not. The simplest form of muscle contraction, skeletal muscle contraction, is the response of the muscle to a single action potential is called twitch. For us, fortunately, most of us, our muscles don't twitch very much. The twitch is simply a transient contraction phase followed by a relaxation phase, and it's a response to a single action potential. Now, what did Dr. Noyen tell you about motor control? Nothing? So when you're trying to lift this thing here, does your motor nerves fire single action potentials? One action. No, they actually, motor nerves fire trains of impulses. So they give you a burst. You can the action potential depending on the task. So you never ever get single twitches in skeletal muscle. Doesn't happen. All contractions are carried out by the motor nerve or the motor axon firing bursts. Maybe 50 action potential. So single twitches are sort of a physiological experimental procedure that you could do that. You can take the motor nerve to some little beast that you anesthetize and stimulate the motor axon with a single pulse, and you get a single action potential, you get a single twitch. So you never get single twitches normally, and not likely abnormally either. So there's the other thing that's kind of interesting is skeletal muscle, complete skeletal muscle only actually, is in skeletal muscle the action potential lasts several milliseconds. Is that reasonable? It's a little slower than a nerve action potential. And how long does a nerve action potential last roughly? Milliseconds? 1 or 2 milliseconds. Skeletal muscle may be about 2. So it has a very fast action potential. And that means that once the action potential is over, you can stimulate that cell again. Because once you're past what is called the refractory period, you can activate the cell again. So there it is possible to initiate a second action potential before the first contraction is completely relaxed. This is known as formation, addition, whatever you want to call it. So, these little arrows are the time that you apply an action potential. And the muscle, of course, the twitch, the single twitch lasts 200 or 300 milliseconds, somewhere under a second, depending on the type of muscle. So you stimulate it here, an action potential, and if this is about 200 milliseconds, then you can probably activate that muscle again. Another action potential. So you get a second twitch. If you do it closer together, then you get the first contraction or the first twitch, and then you get a second twitch. You do the second action, you're summing contraction. And then if you bring these action potentials closer together, still the action potential is over, the refractory period is over, and you can hammer it, hit it again. And of course you get a subcontraction. This contraction is quite different. This one is much bigger than this. So you get a summation effect. And of course if you sort of stimulate the rough, you get this sort of, so this is assuming a nerve fires a burst of about 1, 2, 3, or 15 action potentials. You get a contraction of a motor unit, which looks something like that. And that's the way most the motor units in mammals are activated. You get bursts of action potentials and more or less some contractions. Now, if you only activate one motor unit and you're trying to hold trying to point something out, and you get this nice oscillation, it'd be pretty difficult. People who do cataract operations couldn't do them. They'd have to use a robot. And there are people who have very little tremor. So the idea is you're not activating only one motor unit. You're acting another one, or several which are out of phase. So you may have one that's activated just before and it sort of filters a lot of this stuff out. Because there are people who have very, very little tremor when they try to do some surgery, say cataract operation. Otherwise, yeah, couldn't do it. So basically, the fact that you get several, but not in phase, as you would say. So basically, it means that the contraction or the force that you move or movement of this doesn't shake around. It basically is a fairly smooth contraction. So these kinds of contractions are the ones you get in all the individual motor units. You never get single contraction. It's always trains of action potential. If you really blast it, and this is an experimental procedure, then you get what is known as a complete tetanus. And these kinds of contractions do not occur physiologically. You get somewhere in the middle, some contraction. You never get these. So no oscillations in force, and tetanic contractions, you can get them if you have tetanus. So if you're working in a garden and you cut yourself, and you haven't had your tetanus shot for the last 30 years, you get tetanus, and the toxin from that bacteria will cause tetanic contractions. That means tends to be brutal. Okay, so that's something else. And this is a name which has been given to this complete fusion. And that means that during this complete fusion, the calcium remains very high. inside the cell, and the pumps in the sarcoplasmic reticulum cannot catch up. So the partial one, of course, is trying to catch up, but if we do this, we can't catch up, so calcium remains high. And as I said here, the tetanus occurs because the free calcium remains high, allowing maximum force to be developed. The calcium constants cannot catch up to mediate relaxation. That is not a common thing physiologically. You don't get tetanus unless you have something else wrong with it. And how fast you can stimulate depends on the type of muscle. If it's a fast muscle, the nerve action potentials have to be closer together. Slow muscle that can be further apart. So it depends on how fast this thing works. But that's not of great interest to us. So if you want to generate force, how does it happen? Well, that's kind of a central thing. I don't know if Dr. Moran pointed it out. If you want to lift these are produced not by individual fibers, but by motor units. And since one motor unit, all the muscle fibers in the unit contract together. In man, single twitches do not occur, but contractions are some. Always, you get bursts of action potentials down the motor axon. And for example, for fast motor units, you get bursts of 30 to 60 action potentials per second. For fast motor units, and 10 to 20 action potentials per second for slow motor units. Just a rule of thumb, dado. That's something I'm going to, that's good. Okay, so basically how the changes in force occur. You're trying to lift something. Changes in force are brought about by recruitment of motor units, primarily. You do get changes in frequency, absolutely. You do get kind of situations where you get a little more tetanus or a little less. But most of the force is, for example, if you want to lift 10 grams, you may activate 5 motor units. If you want to apply a 50-gram force, you may activate 20 motor. So it's recruitment of motor units, and that's a central effect. And each unit is some contraction. Motor units during voluntary are activated on the basis of size. So Dr. Monroe told you about the size principle. No? Okay, size principle. You get the smaller motor units first, and they get bigger and bigger and bigger as you increase the force. Small ones first, larger ones next, etc. So that's called the size principle, and it has to do with the central connections. Smaller motor units are innervated by smaller axons, hence you need less synaptic input to drive them. The larger motor units need larger synaptic input because they're bigger. And that's the way the system actually works. Now you don't get changes in frequency. I can't say that that doesn't occur. They do. But that's not a big player. So it's basically recruitment based on size. The other thing about skeletal muscle that's kind of neat, the force, most of your muscles that you use in your body are at an optimal length, somewhere along the line. So basically, the type of force that you can generate depends very crucially on one factor, in addition to calcium and everything else, so a factor on what you would call the actin-myosin overlap. So if you get an optimal myosin overlap, you get the actin-myosin overlap, you get the maximum tension. So, I don't know, got A, B, C, D, none. But anyway, this would be the situation where you get the maximum actin-myosin overlap. These, of course, this is the myosin filament, this is the actin filament, and you get the maximum force in this range here. If you shorten the muscle in this range here, where the actin myosin overlap and the actual actins bump into each other, where the filaments actually slide, then you reduce force. And stretch your muscle out. This is best done in sort of isolated preparations, hard to do in a human. Then as the actin myosin overlap goes to this region where there is none, because the muscle is fairly elastic, you can move, the force will go down progressively. So most muscles in your body, at certain sort of angles and things, have optimal actin myosin overlap. So the force depends on the optimal actin myosin overlap. If it's reduced, you less force, and if it's too short and the actual actins start to bump into each other during shortening, then your force will also go down. It becomes an issue not so much in skeletal muscle, but in cardiac muscle. We'll go through this idea again. So, for your biceps, about 120 degrees is the optimal actin myosin overlap in your bicep muscle. We actually measured it a long time ago. About 120 degrees. So if you stretch it out, you're stretching out the muscle, you're pulling it all apart, so there's less actin myosin overlap. And when you go into this region here, angle below 90, you're going into this where the two actins will bump into. And this sort of demonstrates. The interesting thing about this diagram, this is one of the first real good demonstration that the muscle contracted by the sliding of filaments. And the gentleman at the University of Seattle, and a couple other heavy-duty investigators, he's the first author of the paper, and I think he does the only paper he actually ever published. But he's famous for this. And that's a neat bit of work. Well, that proved that actin filaments were pulled into the H zone by the myosin filaments. Big breakthrough. And again, well, the parents are probably still alive, or alive. Anyway, this is a summary of the different types of muscle, skeletal muscle types. For your information, in case you need it somewhere along the line. A couple of things, skeletal muscle classified into several types of, and it depends on who's doing the classification. This is one classification. Slow twitch, type 1, or fast twitch, type 2. And the twitch is really how fast the muscle contracts into a single action. Some of the muscles, the slow twitch type, and some of the second are reddish in color because they have a protein called myoglobin in them. And that's similar to hemoglobin. And what does hemoglobin do? Or oxygen, right? Well, that's why basically you need it to generate ATP. A white muscle, a white color, they're the ones that primarily use glucose to generate out of lactic acid. And there's telling you the different types of pumps and the different types of release channels are all the same. Calsequestrin is actually the molecule inside the SR that stores the calcium. Calcium sensor is troponin, and they're really different. They're just different isomers of the molecule, that's all. But they work the same. And of course, the pumps are similar. And the myosin heads are slightly different, but they do the same thing. The major difference being, of course, is these rely on glucose. These actually try to generate tension and try to generate ATP like conventional methods first. Okay, so that's, don't memorize the table for your interest. Start on cardiac muscle. Have you been able to copy that off from any class?
Do you have paper I could borrow someone? Like live paper?
Okay, cardiac muscle makes up the heart. General classification, but as I said before, there are different types of cardiac muscle. Depends on what they actually do. So what we do is associate cardiac myocytes. That's what they look like. Go into enzymatic association of this is probably from dog, I guess. No, maybe not. Guinea pig. Some animal. Human ones are very similar. Okay, so then basically looking at it, we kind of look for not comparison to the muscle, nice long cylinders. These are cardiac, individual cardiac muscle cells. So, they're about 100 micrometers in length and about 50 micrometers in width, usually. And they sort of look like they've been cut up by your 7-year-old nephew when he's doing something for a scrapbook or a bird scrapbook. Jagged edge, stuff like that. The thing you should notice is they're banded, the same as skeletal muscle, and they're relatively small. This is a picture showing section with a strip of cardiac muscle. And this is one muscle cell. It then attaches to another muscle cell. So this sort of invaginated region looks like a river going through Vermilion, Alberta, someplace. An A band. There's the I band, the Z line, nice M line. Of course, there's something called GD, which is gas junction, I guess. We'll talk about that. The thing I want to point out here, cardiac muscle cylinders based across their issues, the bands are the same, you have all the baggage. Is this big guy here is the transverse tubular system. And the thing, the striking difference between this and skeletal muscle is that this one is about 10 times the size of the non-skeletal muscle. It's huge. Big hole in the muscle cell. So it said no to large CT. It was much larger, about 10 times. Cardiac muscle and the ventricle and the chambers are made up of muscle cells. And this is sort of an organization, network of cells which are going along from kidney and anastomose. Maybe the cells are short bridges. The regions where the cells join are called intercalated discs. This is an individual cardiac muscle cell. Here's another one. So it's tied to these neighboring cells by intercalated discs, either here or anastomose here, underneath somewhere with another cell. So they're all interconnected. So the big, small cells, and the cells that make up the chambers are made up somewhat like this. These, of course, are capillaries. You can actually see a red blood cell in there. And that's the way cardiac muscle or heart is made up. It's made up of a bunch of small cells. And they're all tied together by these regions called intercalated discs. And some of this stuff we're going to have to deal with in the cardiac muscle. Fine structure of cardiac muscle is the same. Filaments are similar. F-actin, what started out with tropomyosin to identical helices, troponin, TNT. CNI, all the stuff that you have in skeletal muscle is there. Slightly different isomers, but they're all there. The only one I want to make any deal about is the fact that the nebula. Neb is also there as well. Now, there's something else. So nebula is all there, so it has everything. Sorry.